All right. I think we might make a start now. We're a couple of minutes over, so we don't want to take up too much of your time. Can people just verify that they can see my screen? So you should be able to see slide up at the moment. Maybe I've blocked the reactions, but hopefully you can see my screen. Um, anyway, welcome everyone. So this webinar is really just a way to sort of touch base with you about the progress that we've made this year in terms of, how the sample collection has been going, how many samples we've received, how we've sort of been improving our lab methods that we have in-house here to be able to achieve better detection limits, add new targets to our suite as well, and just give you an overall update about what's happening at the moment and what's going to be happening in the coming months. So, my name's Brady. If you haven't been to a webinar before, you might not know my face. So, I'm a PhD student at Deakin University at Burwood, and Pesticide Watch is sort of the project that I lead, but it was originally conceived by my principal supervisor, who's Damien Callahan, and Julie Mondon from the Warrnambool campus. And we also have added a few new members to the team. So we've got Pat Bonney, who's a social scientist at the Burwood campus. And we're also working alongside Renee Webster at the CSIRO at Clayton, who's helping us with some analytical challenges. So, firstly, I'd just like to acknowledge country. So, I'm coming to you today from Wurundjeri country. So, I'd like to pay my respects to the elders past and present and thank them for their custodianship of our freshwater streams. So just a quick summary about what you can expect from today's webinar. Firstly is just some of the background around pesticides. If you didn't come to our sort of introductory webinar for Pesticide Watch, you might not know sort of the context that this project sits within. So we'll just go through that relatively quickly, but hopefully you find it interesting anyway. We'll talk about the progress so far, so how many samples we've received and so on. Then I want to talk specifically about a couple of different analytical things that we've done here, which you wouldn't really know about because we've been doing it all hidden away here. But I think it's pretty exciting and it's definitely worth sharing. So the first thing is we've increased our sensitivity for one of our multi-residue pesticide methods over tenfold from our previous method, which is great. So rather than buying a really top of the line, expensive, brand new mass spec, we've managed to improve our sampling, our sample preparation, which has meant that we've had this massive boost in sensitivity. So I'll tell you about that. And I'll also spend a bit of time telling you how we've implemented a new method for analyzing some really highly polar herbicides, including glyphosate. I think almost every time I've spoken with citizen scientists participating in Pesticide Watch, a question has come up about, can you please analyze glyphosate? And I've had to explain the analytical challenges around it, but we've actually been able to address those and incorporate this method. And we've already analyzed quite a lot of samples for glyphosate at present. So I'll tell you about that. And then we'll go through some preliminary data as well before I start asking you questions as well about how you might like to have this data presented in the future. So firstly, I think it's important just to define our terms. So pesticides can sort of be categorized in a few different ways, but we're using it as an umbrella term like most papers do, as an umbrella term that incorporates things like herbicides, insecticides, fungicides, rodenticides, termiticides, so on. So basically any chemical that's used either to deter or eliminate a living organism. I think it's also important to acknowledge that these chemicals do help us address some really big national challenges. Things like the incursion of the varroa mite into Australia, which is a relatively recent phenomenon, which is likely to harm some of our pollinators. We have fire ants as well. There's a massive fire ant eradication program happening in Queensland at the moment, and this is being addressed with the use of a variety of different pesticides. Pesticides also are sometimes a requirement for biosecurity and for having access to export markets. And of course, there's growing pest and weed resistance to some of these pesticide products, potentially from overuse, which is making us see calls coming for more active constituents to be registered. So pesticides offer a convenient solution to this. They're widely available as well, not just to specialists, but also to people just off the shelf at their local hardware store. However, there is a good side of pesticides, and then of course there is the bad side, which is where we see some pretty detrimental environmental effects. So the first thing we can have are these acute lethal effects, where if a pesticide targets a particular mechanism, for example, an acetylcholine esterase inhibitor, which kills insects, this can potentially affect non-target insects. So we can see these acute lethal effects, and we know that we're seeing some biodiversity loss in Australian aquatic invertebrates as a result of pesticides. What we tend to see more often is pesticides acting as a bit of a stressor alongside a bunch of other chemical contaminants in waterways and having these chronic sublethal effects. So they're affecting organisms in physiological ways, structural, and even behaviorally, they're affecting them. And whether we see these effects or not, and which ones we particularly see, depends on a few different things. I just need to quickly admit someone to this. There we go. Great. Perfect. Sorry. I'm flying solo on this webinar, so I'm having to do a few different jobs at once. So whether we see our acute lethal effects or our chronic sublethal effects, it depends on a few things. So firstly, the mode of action, so how this particular chemical actually works on an organism, the concentration that it exists in a particular stream, the duration of exposure. So is this coming in a big pulse, which is a high concentration, but short in a temporal scale, or is it there at a level which is presenting constant stress to that ecosystem? And then of course it's the sensitivity of individual species to that particular chemical. So we need to learn about all these different things, and this is where monitoring programs are really, really important. So how do we handle this sort of, pesticides are useful and we're used to them, but also have these negative effects? Unfortunately, we often see that there has to be trade-offs, or there appears to have to be trade-offs. For example, a classic one is dimethoate and fruit flies. So dimethoate, of course, very effective on fruit flies. Some parts of Australia don't have that issue and would prefer that their produce arrives treated with dimethoate. And this means that we have a dynamic regulatory landscape. Of course, dimethoate's known to be neurotoxic at a certain concentration, and I don't think there's a better example of a residue in Australia which shows that we have to be dynamic when we get new information. So dimethoate has been in the news since the mid-90s, with public consultation processes, reconsiderations, long chemical reviews that seem to never end. But people expect that when there's new information, we should have a fast and an adequate response to that. And then, of course, we have a sort of unflattering international context on pesticide regulation. So we have this dirty dozen, which is at least 12 different European Union banned pesticides, which are still used in Australia. So we have this really big, complex context around pesticides. What we're trying to really ask is how can we achieve this broader community engagement beyond top-down event-based consultation processes, which is sort of the main interface that we have between the public and the regulator at the moment. So the APVMA may ask for submissions to a particular chemical review, for example. So how can we achieve more active involvement than just those processes? And of course, what's been in the news at the moment? The media in Australia is really highly engaged with pesticides. In just the last year alone, we're seeing a bunch of different headlines popping up. And I think this is the main source that people get their information from around pesticides. So we have links with serious diseases. We have bans, which are brought in and then potentially relaxed and then brought in again over time. We have alternatives entering the market. And then we have controversies around whether these chemicals are actually in our consumer goods or not. So unfortunately, authoritative information, and especially the interpretation of that information, is really scarce in Australia. But there is evident public concern out there about these issues. So we want to try to bridge that gap. Now, pesticide monitoring, having a large amount of ongoing monitoring data for a range of different waterways in Australia is ideal, but there are a bunch of barriers to it that we try to address through Pesticide Watch. The first thing in Australia is that we just have a massive continent here. So there's a vast geographical spread of waterways. And a really interesting one as well is that about 80% of water frontage in Australia is on private land. So having access to those waterways is also really challenging. Then the number of pesticide targets we need to analyze for. There are about 8,000 different active constituents in Australia that are pesticidal. So when we're only analyzing for a dozen or a couple of dozen, we're missing a whole lot of what's really going on. Having a streamlined analysis, which doesn't require huge amounts of solvents, plasticware, analyst time, is very important for a program like this, but also to make sure that we're achieving those environmentally relevant concentrations. A lot of monitoring programs internationally are working in that sort of parts per billion range, which is micrograms per liter. But we're increasingly seeing environmental thresholds be lowered to these sort of nanogram per liter. So these are our parts per trillion, which is a really small amount to imagine. But we need to be meeting that as well with our detection limits. And then finally, one of the biggest barriers is the cost of running a program like this. So we can sort of separate these issues into sampling challenges, analytical challenges, and just a universal challenge that I think a lot of people face when they're trying to do good work. So luckily we can address this sampling challenge. As you all know, you have access to a range of different waterways around Australia. You're actively engaged out there caring for your waterways very, very regularly. And you have great coverage of probably the most important parts of Australia for us to be monitoring for pesticide residues. So if we have a look on the map on the left, aside from the natural areas, which are sort of your purples, pinks, lilacs, and the native vegetation areas, when we start looking at where we're growing our crops, where our human populations are, we have great coverage even just from the Water Watch program alone. So if we're engaging you in this sort of sampling, we're able to get really, really valuable monitoring data. So that was the initial incentive for starting Pesticide Watch. And so Pesticide Watch was this idea of having a really simple, cheap, but robust pesticide monitoring program, which engages a range of different sites around Australia and tries to communicate that data and provide that interpretation as best as we can. So over the sampling campaign, which ran for, well, 12 months, of course, some people started earlier, so they finished earlier. Some people are still collecting at the moment. But we should get most of our samples very soon. We've had 135 sites we have representation in every Australian state. We did miss out on the Northern Territory and the ACT, so we didn't quite get the territories, although we are potentially getting some samples from some groundwater in the Northern Territory very soon, which should be quite interesting for us to analyze alongside our Pesticide Watch collected samples. But we had a range of different participants, and I'm sure you'll see your group represented somewhere in this countdown of different sites, different participants, sorry. So we have, we got WA Department of Water and Environment, we got water watchers, land care groups, schools, universities, traditional owners, all on board with this project. And they're all from a range of different land use regimes as well. So we have agricultural areas, we have urban areas, sort of wetlands, which are really sensitive. So there are all sorts of different land uses that will eventually be able to match up with our exposure assessment. So as you all know, you received a kit and collected those filtered water samples for us, which was hopefully very straightforward for you to do. I hope it didn't take longer than about 5 minutes to collect the sample for us. We tried to keep it as simple as possible, and you froze those samples and sent them in to us. So we have heaps of these 50 ml centrifuge tubes, which are stored frozen at the moment, and we actually do a range of different analyses on these. So it's not just one test that we're doing. So we have 4 different instrumental methods that we do. The first one, we take 2 mils for, and we do a multi-residue pesticide method. So this first one is for our sort of less volatile, or semi-volatile pesticides. And it can detect over 400 different pesticides and measure over 140 of those pesticides. So then we have another one. So the bulk of the sample actually goes towards our more volatile method. So this can detect potentially many different pesticides. So we can do non-targeted analysis with the instrument that we use for this analysis. Haven't done non-targeted analysis yet, but it's something we can always revisit. At the moment, we're doing a more targeted analysis. And we can measure over 200 of those pesticides with this method. Then this is the one that I'm excited about because it's the freshest analysis that we have. And this is for our highly polar herbicides. So glyphosate, AMPA, and glufosinate. So I'm going to talk through the particular analytical challenges around those herbicides and what our solution to that was. And then we're going to also do trace element analysis. So at the moment, next door, I have a student who's organizing, so subsampling all your samples for this trace element analysis, which we'll be able to do before Christmas. So we should be able to run all 1,000 samples by then. So that gives us probably about 15 to 20 mils left in each of your samples. And with that, we're going to retest for salinity, which I'm going to show you in a second is actually really important as we've learnt along this journey over the last year. We'll be testing for pH as well. And we're also hoping to be able to test for nitrates and phosphates from a benchtop microplate reader, which would be really great to do. Those levels, if we get them nice and consistent, might be something we can use as a bit of a proxy for telling us when a pulse of pesticides might be occurring at the same time as a pulse of nitrates and phosphates, which could potentially indicate that these residues are coming from agricultural runoff. So we currently have over 1,000 samples. Yes, we finally crossed the 1,000 samples threshold, which is great. It's a nice milestone to reach. I think we're still probably about 150 or so away from being complete. But yeah, we have the vast majority of them on hand already, and we've analyzed quite a lot of them already as well. So this is the first thing I wanted to tell you about, which is our trap and dilute method. I won't go into huge detail with this, but this is a fully automated sample preparation method, which means we can just put our filtered water that you've collected straight into an instrument vial, and we can load it onto our instrument. And we've done something called a trap and dilute method, which means that rather than directly injecting our sample onto an analytical column and then measuring the amount that's there, we're actually able to inject a huge volume of this. So rather than doing something like 0.1 mils, we're able to do 1.5 mil injections now, which immediately is approximately 15% greater sensitivity, 15 times the sensitivity. So we load a huge amount onto a trap column, and then we reverse the flow and back flush it off that trap column, which then goes onto an analytical column, and then finally for detection, which is, it's probably a little bit, it's quite complicated plumbing actually to get this system up and running. The takeaway is that it's a fully automated sample preparation that happens online as you do an injection. And we get detection limits now in that really low nanograms per liter range. So I think our mean detection limit for this method is about 5 nanograms per liter, which is really environmentally relevant. Those are the sorts of levels that we're expecting to see and that maybe some other labs aren't quite able to achieve. So we'll be submitting our manuscript for publication soon, just on this method, because it's sort of a custom setup and it works really well. It's fully validated now, and it's also been validated alongside our sampling protocol. So this is a really cool thing that we were able to do, and our samples are all going to be analyzed along this particular sample preparation and analysis. Now, there have been some hiccups along the way with this particular instrument. One thing is occasionally receiving an unfiltered sample. So these have presented a challenge. Sometimes samples have particulates which appear to be tannin and really small, but actually end up blocking some of our lines. So we have put in an inline filter, so we can try to remove these before they actually hit the expensive parts of the instrument. Another thing which I told you before was that salinity has become really important to measure as well. And that's because I was, being a more chemistry based rather than environmental based person, I wasn't aware that fresh water doesn't necessarily mean fresh water. And I've been quite surprised at the amount of salt which has been in a really small amount of our samples, but some of them have been really, really quite salty. So injecting a large volume of these salty samples onto our instrument has actually at one point led to the blocking of a part of the instrument where the sample actually enters the mass spectrometer. So this was completely blocked. You can probably just see that the line through the middle of that desolvation line is blocked with white salt. So we subsample all of your samples for our different analyses, and then we check them all for salinity, and then we remove the ones which are too saline for that particular analysis before it actually reaches the instrument. And since we've been doing that, we haven't had any of these sorts of hiccups along the way. But it's been a learning, it's been sort of a learning experience for us as well. So now I want to talk about glyphosate. So you're all really interested in analyzing for glyphosate, and it's so funny that it's a really widely used herbicide. It's available all over the place, and it's used. A huge amount of the herbicide use is just made up by glyphosate use in Australia. But we couldn't actually analyze it alongside our hundreds of other herbicides. So just to give you a bit of background about it, it is a non-selective herbicide. It is the active ingredient in Roundup. It is very extensively used. A bunch of different international authorities, including our own APVMA, the US EPA, and European Food Safety Authority, they all agree that glyphosate is safe for humans. So there's still the odd controversy that comes out about this, with new research coming out all the time. But at the moment, the position is that it is safe for humans. But there's definitely an acknowledgement that there's potential for ecological harm, particularly aquatic plants. When you have a non-selective herbicide and it's used so extensively and entering waterways, of course, there's going to be a potential impact on aquatic plants. So it's definitely worth analyzing for. Now, to give you an idea about the analytical challenge around glyphosate, I'm just going to give you a bit of an analogy about how these instruments actually work. So there's two things we need to do. When we inject a particular sample, we have separation of the different analytes, so the different pesticides in that sample, and then we want to detect each of them. So we can think about separation a bit like a running race. So all these analytes, they all start at the beginning of the track, and then as the race starts, they begin to separate out at different rates. And then eventually, ideally, we have someone waiting at the other end, which is our detector, our mass spectrometer, which is able to take a nice clean picture of each target as they finish that race. So our running track in reality looks a bit like this. So this is a liquid chromatography column. So this is where our separation occurs. And this is sort of what our detector looks like, but we won't have time to go into that. The point is, it's basically like a photographer at the finish line, making sure that we're able to get a clean picture of what pesticide we have. The problem with glyphosate is that it moves really quickly through these columns. It doesn't tend to interact with the column at all. It's basically flying along that racetrack so fast that we're not able to accurately detect it alongside other pesticides. So our solution to this is something called derivatization, which basically is doing this. We're trying to slow down glyphosate by modifying the conditions that it exists in. So then we're able to take a nice clean photo of it. So in reality, this actually looks something more like this. So these are our reaction mechanisms that we have. So here we have glyphosate, AMPA, which is the primary degradation product of glyphosate, and glufosinate, which is another highly polar herbicide. So these are all our really quick herbicides, and these FMOC compounds, so FMOC chloride, can be thought of as those weights that we're putting on it. So we're making it, we're modifying its polarity so that it's moving more slowly through that column. And we're also adding quite a lot of mass to it as well. So by producing this derivative, we're then able to measure that derivative, and that can tell us how much of that particular active ingredient was in there at the beginning. So this process is called derivatization, and yes, I've simplified it, but really that's all it is in essence. So how did we actually go with measuring this in our samples? So we've analyzed 656 of those samples so far, which was all that we had on hand at the time, which is probably 3 or 4 months ago. I think it was maybe September. So we ran 656 samples. We actually didn't detect it that frequently, which was quite interesting. So glyphosate, we measured 66 different samples. We detected AMPA in 35 of those. Glufosinate only popped up in one of those samples from Western Australia. So it's not actually all that bad in terms of the frequency that they're being detected at. The concentrations do vary, but the vast majority of those that we have measured are well below the Australian and New Zealand guidelines for freshwater streams. So seem to be okay at the moment. However, glyphosate is in a bunch of waterways, and we still have many more samples to run. So there's still a lot to learn about what's out there. So we'll put glyphosate aside for the moment and have a look at some of our detection data that we have from the whole network so far. So this is the first half of samples that we ran on our liquid chromatography method. So these are the 30 most frequently detected pesticides that we had in the network. And as you can see, there's a pretty even split between fungicides, herbicides, and insecticides, which I think is interesting because usually in these monitoring programs, fungicides are underrepresented in target lists. So I think by screening for a wide range of different pesticide residues, we've probably captured a little bit more of the story than has been seen before. One interesting one is DEET. So we have detected DEET really frequently, which has been a bit of a surprise until we've started chatting to other people from other labs who often detect DEET as well, but generally say that it's a contaminant from people collecting the samples. We're pretty confident it's not a contaminant. I've definitely collected samples. I've never worn DEET, probably ever. I wear gloves, and I'm picking up quite high levels of DEET in the samples that I collect as well. So we're unlikely to think that this is contamination, and we're seeing it in quite a lot of samples. So this is just shy of 60% of the samples that we've analyzed. We've been able to detect DEET. And we've detected one synergist, so a compound that is used in pesticide products to make them more effective, and also about half a dozen different transformation products. So it is quite overwhelming when you're looking at the whole network and looking at 30 different residues at once. But if we decide to just zoom in on one particular site, it makes a little bit more sense. So here's some data from some sites along Merry Creek. So we have about 6 different Pesticide Watch sites along Merry Creek at different parts of the stream. And as you can see very quickly, we now see that there seems to only be about half a dozen different residues which seem to be dominating that stream, and their breakdown products are also there as well. So let's have a look at them individually. So firstly, we have these triazine herbicides. Now, these are really widely detected in a range of different surface water monitoring studies. So not just in Australia, internationally, they're picked up very often. They're very water soluble, and they tend to stick around quite a while. So for that reason, they're actually banned in the European Union because of their persistence in waterways. And they've been banned for, I think, over 20 years now. And there's also the potential endocrine disrupting effects which have been reported for this class of different herbicides as well. Another different group that we're picking up are these fipronil insecticides. So these are fluorinated compounds that are related to fipronil. So fipronil is common in quite a lot of consumer products, as I'll show you in a second, but they're moderately water soluble, relatively stable once they're in these systems, and they have pretty well demonstrated toxic effects to invertebrates in aquatic systems and also to fish and frogs. So they are on a range of different consumer products. For example, fleas, ticks, and lice treatment for pets, and also some products you can just purchase off the shelf quite easily, like cockroach killer gel, which you can just buy at your hardware store. So it's probably unsurprising that we're seeing these sorts of compounds in urban streams where there's a large human population as well, and a large pet population for that matter. We also have one that we're detecting quite a lot here from the neonicotinoid class of insecticides. So the neonics are highly water-soluble insecticides. They're really thoroughly documented effects that on non-target organisms that have been established, especially pollinators. So these are effectively banned in the European Union. They were withdrawn from sale, I believe, in the mid 2010s. So they've been removed from sale there. And also, I believe that they've flipped and flopped in the UK, but I think now they're not used there as of maybe last year. So Australia, just for this particular active constituent called thiamethoxam, we have 33 different registered products, including 4 household products, which are ant gels. And I went to my local supermarket about a month ago or so and took this photo of a gel that you can just buy straight off the shelf, and its active constituent is thiamethoxam. So again, when we're seeing large human populations like a stream like Merry Creek, where these samples were collected, it's probably unsurprising that we're seeing these sorts of active constituents popping up in that stream. So that's some of the monitoring data that we've got so far. We do have a lot more that we're hoping to share with you in the future, but that gives you a bit of an idea about some of the insights that we're having. One of the things I wanted to talk about, though, is the survey. So many of you may have participated in our survey so far. We've had about 100, I've probably written it down here, actually. It'll pop up in a second, I'm sure. I think we've had about 120 different responses so far. But our idea is to collect a further 1,000 responses for this survey from a nationally representative sample of from an online survey panel, so people who aren't engaged with our project at all, to be able to also measure their responses to this. So you will have noticed if you go through the survey, we're interested in looking at people's risk perceptions, their beliefs, trust positions especially, and their own personal use and purchasing behavior around pesticides. So our sort of model for how we're analyzing this data is to look at people's attitudes around pesticides. So things like their emotions around this, their self-efficacy, perceived knowledge, also their confidence in their own knowledge, and how they process this to develop a risk perception. And then mediated by sociodemographic variables, see how this influences people's own risk reduction and their mitigation behaviors. So what products are people choosing? Are they engaging with authorities, and how do they seek information? And what products do people actually own? Do they use them frequently or infrequently? Where do they store them when they're done with them? So these sorts of questions we think are really important. And some of the reason why we think they're important are because there are massive gaps around this interface between regulators and the community. So we want to understand, what do Australians currently know about the potential risks around pesticides? Is there a knowledge gap that we need to fill? Is this issue even important to Australians? And if it is, who's it the most important to? Who's it the least important to? And does engagement with a community science project like ours increase knowledge or shift risk perceptions at all? So far we have, there it is, 122 responses, which we're going to boost with a further 1,000 very soon. And there have been some really diverse opinions in there. Some strong opinions on either side from looking at the preliminary data. And there was an optional comment section, which is a very interesting thing to read. Of course, all of these are anonymous when we have no idea who's responding to these, but it's very interesting to see people's responses on either end of the fence when it comes to pesticides. So I think that the outcome of a survey like this is going to be really valuable for a range of stakeholders, including the development of more projects like ours to sort of help with community engagement. So it was a really extensive survey, as you will have noticed. It goes for about 15 minutes to 20 minutes, depending on how long it took and how many pesticides you owned. But I took this one question out because I thought it was quite interesting for the context of a project like ours. So this is a Likert question, strongly disagree to strongly agree. And I thought this first question was really interesting. So I believe that stopping pesticides from entering the environment is a shared responsibility between government, industry, and community. Overwhelmingly, we have strongly agreed to this one. So it does seem that there is an acknowledgement that it's not just a government or industry problem. Community has an active role in this in terms of, maybe they want to be more engaged, maybe the people who responded to the survey see that there could be more of a role for the community to be engaged in things like monitoring and making decisions around the registration of different chemical residues. I think this one here was also quite telling. I believe that these days pesticides are safe. We got quite a lot of strongly disagrees, and generally we're on the disagree side of this. So people do perceive that there is a problem with pesticides and that these are inherently harmful chemicals. And then this one was one that I shared when I had a meeting with the EPA very recently. I trust that local authorities adequately monitor pesticide contamination in my area. Strongly disagree. So people do perceive there to be a strong gap here, that their local areas aren't adequately monitored for pesticides. So a project like ours that can be far-reaching is something that likely should have longevity and can fill this gap. So this is really, really interesting. But as I said, it's an extensive survey, so there's many more insights to be had from that as well. So a quick update as to what's happening right at the moment. We're analyzing the remaining samples for multi-residue pesticides, so that LC data that I was showing you. We've also got our glyphosate, AMPA, and glufosinate analyses, which I'm hoping to squeeze in before Christmas. If not, it'll be happening in January. We're also, as you may have seen in the newsletter that I sent out, conducting an interlab validation with Eurofins in Melbourne. So Eurofins is the biggest environmental commercial lab that's out there in Australia. So they've agreed to help us just in kind because they see the value of a project like ours. So what happens is we collect a sample, they take a portion, we take a portion, we analyze them for pesticides, and hopefully our numbers agree. So we're currently undergoing that process with Eurofins with their support, which is really great. And that will add a huge amount of weight to the data that we end up sharing at the end of this project. Also, we're going to be doing the trace element analysis. As I said, I've got a student next door who's currently subsampling these for trace element analysis, which should be happening either next week or the week after for 1,000 different samples. And then also I'm exploring some more gas chromatography, so our more volatile pesticide analysis methods at the CSIRO at the moment. And then finally, in terms of the survey, we are preparing to go through that online survey panel and boost the numbers for that survey. So we should have, hopefully, a very big sample size, which can tell us a lot about the social context that Pesticide Watch sits within. Now, this is the part where we start, getting feedback from you, hopefully. Hopefully I can figure out how to enable the chat settings in here. Because I do want to hear from you. If that doesn't work at the end of this, please email me with your ideas and your insights, because what I really want to do is hear how you want this data presented to you at the end of this project. So we are looking at the finish line. We don't have all the raw data yet. We haven't done the full data analysis of what it all means, but we will have that probably quicker than we think. So we need to think about how you would find it most meaningful to have this data presented. So I'm going to tell you a couple of different options that I've come up with, but please feel free to add to these. If you don't have my email already, it'll be at the end of this presentation, so please take it down and you can write at length about what you think we should do with it. But here's a couple of options. First, interactive maps. So this is definitely the first idea that came to my head when I thought about how to present these. We have an example of this being done in Queensland as part of the Reef 2050 project, where they've got some pesticide data from a range of different sites. So this is hosted online. You can log in, you can click on a particular site, and you'll be given, let's see if this will work. There we go. You can get a different plot for each different residue that they analyzed for at that particular site over time. And it'll tell you the concentration over time. It'll tell you where it was detected and the level that it was detected at. And it will also include things like the guideline values for that particular residue. So I think this is quite a good way to do it. But I like plots, and visualizing it with a map seems like a really cool way of doing it. There's another good example of this, which is the Map My Environment, where EPA Victoria hosts some of their trace element data on this platform. Not actually sure who hosts it. I don't think it's EPA because it is a national Map My Environment portal. Um, but they have a similar sort of thing. They've got a bit of a traffic light system going on with these to tell you where there's worse contamination and where it's not so bad. They tell you, the number of samples. You can toggle between different contaminants and different matrices, and you can look at the average concentration of these different residues there as well. So these are a couple of different examples of interactive maps, but perhaps this isn't the way to go. There are other options as well. So here are some that came to mind. You'll remember when we did this pilot study for Pesticide Watch a couple of years ago, if you were involved, we had a short report. I think it was about 5 or 6 pages with just some quick facts about what we found. And it was, written in a way that was hopefully pretty accessible. And we included a bunch of plots in there as well. So we could do a report style thing. The other thing we could do is try to write articles if you find that more helpful, maybe in places like the conversation. And then we also have videos. If you're more of a video person, we can actually try to put together videos which helps explain and interpret some of the data that we've got. So if that's something that you think would be helpful, please let us know and we can look down that path as well. So I'll quickly wrap up this presentation and then hopefully we can have some questions and ideas shared in the chat. But where to next? The first thing is please stay engaged with our project. Please share ideas, articles, questions, stories. Lots of people have done this already. With my email, people are sharing lots of interesting information with me and new findings and newspaper articles and things. Please stay engaged. I'll respond to those emails when they come in. I'm not going to delete them. I'm very interested to hear how people are engaging with this issue. The next thing, data is on the way, so please stay up to date. Check the Facebook page now and then. If I send out an email, it could be that the data's about to be put up somewhere. So we are finally acquiring that last data. So hopefully it won't be too long until we're able to host it online in some form. The other thing is, if you liked participating in our project, and especially the sample collection, and you find some of this data interpretation helpful, please spread the word. We are starting to think about the longevity of a project like this. Some industry partners have thought about, maybe we could collaborate on this and make this a bigger thing and expand it. So if you found it helpful, yeah, maybe chat to your, Water Watch coordinator or something and say that you found it useful. It's always helpful. And ultimately, we're doing this all to achieve these better outcomes for waterways. So I think that brings us to the end of my update. Yeah. If you haven't got my email address, write it down. We've also got the Facebook page linked there, and the Pesticide Watch website, which is hosted on the Deacon's portal, listed there. But yeah, also just a thanks to Eurofins for helping us with that interlab validation, and Slyro for letting us use some of their fancy equipment. If you haven't completed the survey yet, you can do that via the QR code that's there, or you can access that via the Facebook page or the Pesticide Watch website, or you can just email me and I can send you the link for that. So I'll give you a couple of seconds to do that if you want to scan that QR code or write down that email. Otherwise, I might quickly jump off and enable the chat to see if there's any comments or ideas in there about data presentation.